Hi, I'm Natalie Douglas and this is my video on inherited disease. Today we're going to discuss the roles of DNA, mRNA, tRNA and ribosomes within protein synthesis. We'll discuss the inheritance of gender, the inheritance of disease, genetic screening and process, and the moral and ethical issues around DNA recombinant technology. There is a lot of information on all of the slides should you need to revisit. Synthesis is the process which our cells make proteins. This is done using an assembly of amino acids, also known as polypeptides, which then fold into three-dimensional shapes to become functional proteins. Protein synthesis is a process which is really important for cell structure and function, as proteins play an important role for all biological processes within our entire body. Protein synthesis is widely known as the central dogma of molecular biology. The process relies heavily on tRNA molecules to bring the correct amount of amino acids to the ribosomes based on a codon sequence. We'll discuss more on protein synthesis and the processes within this, transcription, translation. We'll also discuss DNA, mRNA, tRNA, and ribosomes and their roles within protein synthesis next. Is the first stage of protein synthesis. This is where a specific segment of DNA is copied into messenger RNA, mRNA, by the enzyme polymerase. Initiation is where the RNA polymerase binds to a promoter. Elongation then begins. RNA polymerase makes its way along the DNA strand, adding nucleotides to the ever-growing mRNA chain. Thymine in the DNA is then laced by uracil in the RNA, which then leads us nicely on to termination. Transcription carries on until the RNA polymerase reaches the termination signal. At that point, a newly synthesized mRNA strand is released, and transcription is complete. On to translation. Translation, this is stage 2 of protein synthesis. It's where ribosomes read the mRNA to assemble amino acids into proteins. Steps involved include initiation, elongation, and termination. In initiation, the mRNA binds to a small section of the ribosomes. The ribosome then searches the mRNA until it finds a start codon, which is usually coded AUG. This then signals the start of the protein coding sequence. We then move on to elongation, where tRNA molecules transfer a specific amino acid and bind to the ribosome. tRNA anticodon then couples with a corresponding mRNA codon, allowing the ribosome to catalyze the formation of the peptide bonds between the amino acids, which then elongates the polypeptide chain. Moving on to termination. Translation then continues until it reaches a stop codon, usually coded UAA, UAG, or UGA, which then signals the end of the protein synthesis process. The completed polypeptide chain is then released from the ribosome. In a nutshell, DNA serves as the blueprint for building proteins. The main role of DNA is to transport genetic information and oppress the process of protein synthesis. DNA produces a set of instructions to instruct the cell to produce proteins. mRNA, also known as messenger RNA, is a type of single-stranded RNA incorporated in the protein synthesis process. mRNA is built up of a DNA template during the process we just heard about, transcription. The role of mRNA is to carry protein information from the DNA in a cell's nucleus to the cell's cytoplasm, where the protein making reads the sequence and translates each 3-base codon into its corresponding amino acid. tRNA, also known as transfer RNA, is a small RNA molecule that plays an important role within protein synthesis. Transfer RNA serves as the link between the messenger RNA molecule and the growing chain of amino acids that make up the protein. In summary, tRNA is a translator. It reads codons and delivers them to the correct amino acids so that the protein chain can grow. Ribosomes. Ribosomes are built up from ribosomal RNA and proteins. Ribosomes are responsible for protein synthesis. They translate genetic information into proteins that are ready to function. They are sites at which information carried in the genetic coding is converted into protein molecules. Ribosomal molecules of mRNA decide the order of the transfer molecules that are bound to the nucleotide codons. Ribosomes play a central role in molecular biology for all living cells, so pretty important. When protein synthesis goes wrong. Protein synthesis can and does go wrong, as we have already discussed the importance of proteins within our everyday functions, and we now realise that they are absolutely critical for a healthy lifestyle. Transcription errors. They're mistakes that are made during the transcription of DNA and mRNA, causing incorrect sequences to be carried forward. Translation errors. Translating information incorrectly, for example, misreading the codons leading to incorrect amino acids being inserted into the polypeptide chain. Point mutations, which are alterations of the nucleotides, which include insertion, deletion, causing the protein to be drastically altered. More on this topic next. Point mutations. Point mutations are a type of mutation where a single nucleotide base in the DNA or RNA sequence is altered. This can be done by submission, deletion, insertion of the nucleotide. There are two types of point mutation: transition mutations and transversion mutations. In transmission mutations, these are caused by one of the pyrimidine base being replaced by another pyrimidine base, or when a purine base is replaced by another purine. Transversion mutations occur when a purine base swaps for a pyrimidine base. Insertion is when more nucleotide bases are being added to the DNA sequence, whereas deletion is where one or more nucleotide bases are removed from the DNA sequence. Point mutations change how the proteins function. They can be caused totally spontaneously or can also be caused by external or environmental factors such as X-rays and UV radiation. The inheritance of disease, and particularly inheritance of cystic fibrosis. Cystic fibrosis is a genetically inherited disease that affects the protein in our body and the protein it produces. This protein affects the body's tissues, cells, and glands that produce our body's mucus and sweat. Genetically, this has happened because the sufferer has received 2 copies of a recessive gene. This in hand causes mutations in the cystic fibrosis gene. This means their mother and father both carried at least 1 copy of the CFTR gene. Furthermore, those with 1 copy of the defective gene are known as the carriers. They do not develop cystic fibrosis because they have a dominant gene that dominates and causes the CFTR protein to be processed correctly within the body, leaving the individual with minor, if not any, effects. The inheritance of gender. Biological sex determination is inherited by using a combination of our genetic information which is carried on our chromosomes. If a Y chromosome is being carried, this would encourage the development of male reproductive organs, whereas vice versa, the absence of a Y chromosome would lead to female reproductive organs developing. The X and the Y chromosomes determine our sex. The Y chromosome contains the SRY gene, which triggers the development of male testes. Without the presence of the Y chromosome, the body would revert to female, which would lead to the development of ovaries. Only sperm carries either an X or a Y chromosome, whereas eggs carry only an X chromosome, meaning males will always determine a baby's gender. The inheritance of sex-linked conditions. In X-linked recessive disorders, males who inherit the variant typically have the disease due to the fact they only have one X chromosome. In contrast, females who possess two X chromosomes, along with the presence of a second non-pathogenic allele, usually protects them from the full disease exposure, meaning most of the time females remain unaffected or only mildly affected. Sex-linked conditions are located on the X or Y genes. These present most commonly on the X chromosome. The inheritance pattern relies strongly on if the X-linked recessive or X-linked dominant. X-linked recessive are males with one X chromosome. A single pathogenic variant is enough to trigger a condition, whereas the X-linked dominant, only one pathogenic variant is needed for the condition to be present in both males and females. An example of a sex-linked condition is haemophilia. This is an X-linked recessive disorder. It initially affects males who carry only the one X chromosome, who can then pass to females, which just discussed could be unaffected or only mildly affected. Chromosomal abnormalities in an inherited disease. A chromosomal abnormality is a change within a chromosome that leads to the inheritance of a genetic disorder. These changes can occur in two ways: numerical abnormalities, where an individual is missing one of the chromosomes from a pair. This is called monosomy, whereas when an individual has more than two chromosomes, this is called trisomy. An example of an inherited disease caused by numerical abnormality is Down syndrome. This is where an individual has 3 copies of chromosome 21 rather than 2. Structural abnormalities, where a chromosome structure is altered in one of the following ways: deletion, where a part of the chromosome is removed or missing; duplication, where a part of the chromosome is duplicated, resulting in too much genetic information; translocation, a proportion of the chromosome is moved to another chromosome; inversion, a part of the chromosome is broken off, twisted, and reattached, resulting in inverted genetic information; rings, a part of the chromosome has broken away, resulting in the formation of a ring. Chromosome abnormalities often occur in the process of mitosis, and as previously discussed, can be completely spontaneous or inherited. Genetic screening. Blood tests are taken from the individual. The geneticists then get to work on the detailed genome sequencing, which is a process that looks at the whole genetic makeup of an individual. Your genome is made up of thousands of sequences of genetic information. DNA is extracted from the blood sample and amplified using a polymerase chain reaction. Increased DNA samples are then analysed to find a sequence of nucleotides and help identify pathogenic variances. Sequencing can also compare genetic information to others and maybe even family members or parents to help identify where the sequence could have gone wrong. This will help to diagnose the individual. An advantage of using genome sequencing is nucleotide variants, so small insertions and gene deletions can be detected with high accuracy and quite easily using this method. On the other hand, a limitation of genome sequencing is a large number of the identified variants is a significant challenge and very time-consuming. Recombinant DNA technology. Recombinant DNA technology involves combining genetic information from 2 different organisms to create new genetic information, or genetic sequences. This is most helpful in medicine, agriculture, and even biotechnology. Outcome from this is recombinant DNA, which is made to produce proteins and change an organism from the original genetic information for a range of different reasons. The moral issues when using recombinant DNA technology in medicine include the worry that this could change a human's identity. It can create diseases, as well as concerns over the technology being misused and data being stolen from individuals. From an agriculture perspective, the moral issues are the concerns over altering the nutritional values of crops, potential health risks, and environmental impact. In the treatment of diseases such as cystic fibrosis, the DNA recombinant technology helps by correcting the genetic information that has caused the disease. It uses integrating and non-integrating methods. Non-integrating gene therapy does not permanently change the genome, whereas on the other hand, integrating gene therapy works to alter the genome more permanently by delivering new genetic information into the cell. In contrast, a disadvantage of integrating gene therapy is that there isn't much control over where the new CTFR gene is inserted into the cell, meaning if it's inserted into a critical selection of the cell, this could increase the risk of cancer. Thank you for watching my video. There is a lot more information on the slides should you need it.